Hello and welcome to today's webinar organised by The Pathologist and Illumina. My name is Fedra Pavlou, editor of The Pathologist, and it's my great pleasure to be the moderator of today's online event, which will be looking at next-generation sequencing as an enabling technology for clinical pathology laboratories. Please allow me to introduce today's speaker, Dr. Rachel Butler, who is the head of the All-Wales Genetic Diagnostic Laboratory in the UK, a laboratory that provides diagnostic analyses for over 50 genetic disorders, handling over 20,000 samples each year. Rachel is actively developing the lab's portfolio of molecular assays for solid tumours in response to clinical demand and working with the clinical teams to determine the validity and utility of these markers. The lab is one of the Cancer Research UK's stratified medicine technology hubs, and Rachel is a grant holder for a number of stratified medicine clinical trials. A fellow of the Royal College of Pathologists in Molecular Genetics, Rachel has a keen interest in external quality assessment and is the current chair of the Molecular Pathology Specialist Advisory Group for the UK's National External Quality Assessment Service. She's also a representative on the Royal College of Pathologists Working Group on Molecular Pathology and the Genetics Specialist Advisory Group. Today, Rachel will discuss her experience of implementing next-generation sequencing into a clinical molecular genetics practice at the All-Wales Genetic Diagnostic Laboratory. Without further ado, let me get today's webinar underway by passing you on to Dr. Rachel Butler. Rachel, over to you.
Thanks, Fedra, and welcome, everybody. So as Fedra says, I'm going to be telling you all about the way that we use next-generation sequencing in my lab for the delivery of services for solid tumours. So I've put together a list of the things that I'm hoping that we're going to cover, and this will hopefully give you some ideas about the kind of toolkits and things that you need to think about if you're delivering and developing next-generation sequencing services within this field. So let's get started. So I probably don't need to introduce to many of you what we mean by personalised medicine or stratified medicine. But this is the way in which we take a group of patients, as you see in the middle there, in all kinds of different colours, who basically have the same clinical diagnosis. So in this case, they all have cancer. Now, traditionally, we'd have treated this group of patients all with the same prescription, shall we say, or we'd have given them the same treatment. And that would probably have been either chemotherapy or radiotherapy. But now we're actually able to start teasing out that group of patients to be able to say, well, which patients do we think are going to predict to different types of therapy? Which therapy or treatment is the best option for a particular patient? And we can do this using clinical phenotype. We can use it, and we can do it using various different biomarkers. And the ones we're going to be talking about today are genetic or molecular biomarkers. So as an example of this, I've taken in two examples from lung cancer, which are really well known. So we have two biomarkers here. The one at the top is the EML4-ALK gene fusion, and at the bottom we have EGFR mutations. Both of these types of mutations you'll find in non-small cell lung cancer patients. They're not very frequent. But what we can see is that if we select for patients who have either the EML4-ALK gene fusion or the EGFR mutation, we can see that those patients respond much better to the treatment that we're actually able to give them. So in the case of EGFR mutations, those patients respond much better to EGFR TKIs. So hopefully you'll all be quite familiar with these examples. And as a result, these particular drugs and their therapies have now been licensed for use in the UK and Europe as well. But in order to deliver those services, we've had to develop operational pipelines in which we can take tumour samples from the patient, extract DNA, perform these molecular analyses, so in this case for EGFR mutations or for the ALK gene fusions, and get a result back to the treating oncologist, all within a meaningful clinical time frame. So what I've shown on this next slide is our molecular strategy, the one that we use here in Wales, but I'm sure it's very similar in labs all across the world. So a patient is seen by the oncologist or the treating physician. A sample is taken, so a biopsy sample, a tumour sample. That sample is sent to the local pathology lab where they determine that, yes, the patient does have cancer. And they also do something for us, which is to assess the sample, and we'll talk about this later on in the talk, to determine that that sample truly, that they're then sending to us for molecular analysis, truly contained tumour cells. Having received the sample, we extract DNA, we perform EGFR, in this case molecular testing, and fax the report back to the referring oncologist so that they can make appropriate treatment decisions. And the whole of this process takes 5 to 10 working days, so hopefully within an appropriate time frame for that particular patient. Oh, we'll get these slides moving. So again, we've got the example here of EGFR mutation positive patients being the ones that respond to EGFR TKIs. And if we look at the scans for this patient who has an EGFR mutation, on the left-hand side you see the before scan, and on the right-hand side the after scan, which shows that the tumour in this case has clearly been removed almost completely. So a really good response for these patients who have the right molecular profile. So we've talked about lung cancer. There's lots of other examples in this same field. So in this table we've got a list of different tumour types and also the molecular biomarkers that we're now using in order to inform our treatment decisions. So that's great, that's fine. We're also using a whole series of different molecular technologies to detect these mutations. So on the right-hand side, that's actually a series of different technologies that are used in the UK for detecting EGFR mutations, taken from a recent UK NEAT-QAS round. So you can see that many labs are choosing to use different technologies. And technologies like COBAS and Seroscreen, which directly identify the EGFR mutations, are really quite popular. NGS is not so popular. However, things are now changing. So if we go back to our example of non-small cell lung cancer again, and we have a pie chart on this slide which shows the breakdown of mutations that you might find in a patient with non-small cell lung cancer, you can see that if we just test for EGFR mutations in red and the EML4 ALK mutations in yellow, we're missing an awful lot of other mutations which could potentially actually also determine the treatment or the treatment options for those patients. We might not actually have treatment options available just yet, but we might have clinical trials which the patient could enter should we know their particular molecular makeup. So if we take a trial like this one, this trial is called FOCUS4. It's actually a trial in colorectal cancer. We're selecting patients on the basis of a whole series of different biomarkers that you see under A, B, C, D, and N. And then based upon those particular molecular biomarkers, the patients that will then receive different treatments. And this particular trial is expanding all of the time, and we're adding on additional arms. Here we have E and F, which show even more molecular biomarkers and more treatments. So this is very much an evolving field. We need to have molecular technologies that can detect an increasing number of different mutations in different genes as the research moves along and tells us that we ought to be analysing for them. And another trial, which is quite similar, a UK trial, is a trial called the UK Matrix trial. This is a trial for lung cancer, and on the left-hand side we have a series of molecular aberrations, so changes in a whole load of different genes and different mutational mechanisms as well. And depending upon which gene a mutation is found in, the patient will be given drug 1 to 8, and they'll be randomized to that arm of the Matrix trial. So I hope what I'm showing is that, in fact, there's an increasing need now, rather than to just test for a set of mutations in one gene, such as EGFR, we're now being asked to test for a series of mutations in a whole series of genes, so maybe 10, 20 genes at a time. And therefore, our molecular technologies that we have been using for a couple of years are maybe not the best choice anymore. So if we think about these choices, we can either test using a gene panel on the left-hand side, or we can test in series using a series of the molecular technologies such as pyrosequencing, real-time PCR, FISH. So what are the pros and cons of the two? Well, if we look at single gene tests, what are the problems there? Well, we've got to provide multiple single gene tests, as we've just shown from those couple of clinical trials. There's lots of different genes and mutations we now need to look at. Now, if we're performing more tests, we're going to require more DNA. Now, from some of the biopsy samples that we're all used to handling, we know that we can't get a great deal of DNA out of those samples. The other problem with performing many tests in series is that that's going to increase the turnaround time of our testing for the patient, and therefore it's going to take us longer to get that result ready for the patient in time for their treatment. And the alternative on the left-hand side is to test using a gene panel technology. So this is using next-generation sequencing. We'll talk about this as we go through the rest of the presentation. So we can test simultaneously for a whole series of different genes and mutations. So it's one single operational process for all gene targets. So in terms of your lab staff time, that's got to be a lot better because they're only actually effectively running one assay. That, therefore, is going to reduce your costs overall. Well, it ought to reduce your costs overall, even though next-generation sequencing on paper is more expensive than single-gene assays. And the other benefit, which I haven't got on this slide, is in fact that we don't need as much DNA because, yes, you might need more DNA for a next-generation sequencing assay, but not as much as if you were going to do 5 or 6 tests in series.
Great, thanks Rachel. So this brings us to our very first polling question. So could you answer A, B, C or D to the following question? Which of these statements might reflect the use of NGS in your institute's oncology practice? Answer A, used only in the context of clinical trials. B, used in routine clinical practice and in clinical trials. C, familiar with NGS but not used it because it is not available to our laboratory. Or D, never heard of NGS being used in oncology practice. Again, to repeat, the question is, which of these statements might reflect the use of NGS in your institute's oncology practice? Please answer A, B, C or D now. That's great, thank you. So, interestingly, we've got answer A, used only in the context of clinical trials at 20%, used in routine clinical practice and in clinical trials at 35.6%, familiar with NGS but not used it because it is not available to our laboratory, 40%, so the highest number of all, or never heard of NGS being used in oncology practice, we have 4.4%. Over to you, Rachel. What do you think of those?
Thank you. Well, I think that's probably pretty much as I would expect, and certainly from my point of view, I would have answered B on this, so used in routine clinical practice and in clinical trials. But I think perhaps it does represent a movement towards the use of NGS, but maybe not in prime time yet. So we're still quite happy if we only have to test for a RAS mutation, we'd still use one of our PyroSync sequencing or real-time PCR options. And maybe we don't have the clinical utility of a large number of markers yet for us to justify in our own laboratories the movement to NGS, which requires the additional validation and the additional costs. But it is really good to see that laboratories are beginning to move towards and adopt NGS within their laboratories, certainly for clinical trials. So a good move and very positive for the future. So this is the way that I see us using gene panel testing in the clinic for our clinical trials as well. So along the top, we have our current pathway of a diagnostic biopsy being taken. We perform our single gene tests for us here in the UK in the NHS, and the patient will be given a treatment on the basis of that. And then a patient may be consented for a clinical trial, which means that that sample that we had, the FFPE sample or maybe the DNA sample, is then sent off for some study for a clinical trial for further molecular testing. So in this case, for a BRAF test. And the patient then may be recruited to that clinical trial if they have their appropriate molecular genotype. So we're kind of scrapping that process now by using NGS and saying, well, can we do a better job in taking this collaborative pre-screening route, whereby a patient receives generic consent for either NHS treatment or a clinical trial. We perform a panel test using next-generation sequencing for a whole series of genes and mutations. And then depending upon the result, the patient may receive standard clinical NHS treatment, or they may be recruited to one of our clinical studies. But we've got all of the results ready and available in one place, one stop. That means we can share the costs between all the parties. It's clearly much quicker. That's got to be better for patients. We're going to be performing fewer rebiopsies because, in fact, we've got enough sample to perform all those tests. And it means that we can test even for rare mutations, as you've seen in some of those smaller parts of the pie chart. And that means if we take lung cancer as an example again, we go back to having EGFR mutations and EML4 ALK gene fusions being our approved and licensed biomarkers along with treatments. They're fine, we know what we're doing with those, but at the same time we can detect mutations in KRAS or NRAS or MET amplifications, which should mean that patients can also enter clinical trials. And we do that all through our next generation sequencer on the left-hand side, appropriate bioinformatic analysis to be able to detect all of those genes and mutations simultaneously. Can I have the next slide, please? Okay, assuming we're on it, I can't see it. So what are the things that we need to be thinking about when we're introducing next-generation sequencing for solid tumours, and what are we doing about tackling them? And I'm going to go through each of these different issues now in turn. So the major problems are that we don't have very much tissue, and what we have is rubbish. It's not like working with good quality DNA. There's a constant change of the genes and mutations that we need to be analysing for, so we need to be thinking about that as well and have the flexibility to change our approach. We've got to validate whichever molecular methods we use in our labs. We also need to be able to interpret the variants that we find, because as soon as we start doing large amounts of sequencing, we're going to start detecting changes that maybe we're not quite sure how to manage. The problems with tumours is that they demonstrate heterogeneity, so that's a problem we need to think about. And at the end of the day, we've got to have acceptable failure rates. We can't select a methodology which is going to be failing all of the time, because that's no good for the clinical care of our patients. So on the next slide, I've put down the aims of what we're trying to achieve through these next-generation sequencing services, or any molecular services to support a clinical trial or clinical diagnostics. So we need to be able to have confidence that we can cover all the genes and mutations which may be valid, or at least a good 95% of them. This method has got to work on very small amounts of DNA and very poor quality DNA. And we want the assay to pass on greater than 90% of samples. We don't want to spend our whole time having to do additional assays because a particular sample hasn't worked, or performing something called gap filling by next generation sequencing. So how are we going to tackle some of these? Well, let's start off with the sample on the next slide. So the real problem with FFPE is, so paraffin 6 samples, is we're going to get minimal amounts of tissue are going to give us minimal amounts of DNA. The next problem is that by fixing the tissue, we introduce something known as fixation artefacts. So if you look on the bottom left-hand side of this slide, you'll see this is actually a screenshot of some next-generation sequencing, where each line actually represents a different sequencing read. And what you see in this particular screenshot is it's a very dotty picture, lots and lots of different spots of red and yellow and green colours, which shows us that, well, it looks like we have a very low level of mutations, but none of them happening at the same position in the same sample at the same time. And what this actually reflects is artefacts which have been introduced through the fixation or other process to that particular sample. And it's not the kind of sequencing that we want to see, because it makes it very difficult to interpret. So that's a real problem we've got to think about. The other problem on the right-hand side is that when we get degraded and fragmented DNA, we're less able to create good library preparations. So at the top, we have a poor library prep, which has come from the paraffin 6 sample, and at the bottom, we have a good library prep, which has actually just come from lymphocyte DNA, so much better. So we've got to think about improving those. So on the next slide, you'll see some of the ways that we can some of the methods that we can use to try and improve our samples. And the first thing that we've done in Wales is to work really closely with our pathology colleagues to ensure that we have minimum quality requirements for exactly how much tissue are we going to receive. So we need to ensure that we receive enough slides. So we said 6 slides of 10 micron thickness. So this might vary depending upon the different application. We then need to ensure that there are plenty of cells on that slide. So plenty of cells that we're actually able to extract DNA from. We also prefer to have samples where we've got greater than 50% neoplastic cell content, although we accept that this is not always possible. And once we've extracted DNA, we always measure that DNA to ensure we've got sufficient DNA to actually perform our next generation sequencing. And we measure this with either the qubit or the bioanalyzer because they're good and sensitive. So on the next slide, what I've tried to show is the way that we're working with our pathologists to improve the samples that they're able to send to us and provide for us. So what I've tried to put together here is a cartoon of a piece of tissue within a paraffin block. So that's like the big blob in the middle. And then what routinely happens in a pathology lab is that the pathologist will take a series of sections, so from the left-hand end there, initially to perform an H&E to actually say, well, is there tumour within this sample or not? Now, say on that first section, the pathologist decided that it wasn't a good enough section and they wanted to go back in another one, they would cut through several more sections of the block before they take a second H&E. On the second H&E, they're able to determine that tumour is present, and then the pathology lab might perform a whole series of stains in order to determine the histology of the tumour. And then a further H&E is performed in order to confirm that they're still looking at the right region of the tumour sample. Now, generally, in many laboratories, that block would then be put away. It will be archived until somebody like me or the referring oncologist said, can we actually have that block back out so that we can now perform molecular testing? At that point, the block has to be refaced. So basically, it has to be set up on the microtome so we actually get a clean face in parallel to the microtome. So we can get some more sections cut. Another H&E is performed, again, to ensure that we're looking at the right bit of tumour before samples are finally cut for molecular testing. And I hope what I've been able to show there is that, in fact, often the sample which is used for molecular testing is really the dregs of the sample if we're lucky. And it would be much better if we could remove some of the waste, particularly from the middle of the block, the scrolls and sections that are actually just go into the bin. So if we can persuade our pathology colleagues to cut much earlier in the process for molecular testing, or even better, provide the molecular testing within the pathology laboratory so it's a truly joined up process, then hopefully we'll be able to provide far better samples. So moving on to the next slide, we can now start thinking about, well, what do we actually want to include in our gene panel? We know we've got reasonable quality of DNA, so what do we want to test for? And is there an ultimate panel size? Do we want to test for 5 genes or 500 genes? You know, we could sequence a whole lot. We could do a whole genome sequencing, but I'm not sure that that's a very sensible thing to do for the kind of applications that we're talking about. We know that our panel design is going to be limited by the fact that we're using paraffin-fixed tissue because our amplicon size has to be shortened. We do need to think about what kind of application are we using. So if we just want to use this gene panel just to look at clinical applications, that we may choose to only analyse 4 or 5 genes. Whereas if we want to be analysing on more of a research basis, we might want to look at 100 genes or even 200 genes. This is all going to impact upon your budget. The more genes that you analyse, the more it's going to cost you. And the other thing to think about is, are there some genes that you really don't want to know about? So, for instance, do you really want to know a lung cancer patient's BRCA status? So, do they have a mutation, which may be a germline mutation in BRCA1 or 2? So are there certain genes which you think, right, I really don't want them on my panel, or if they're on the panel, I don't want to see the results of them? And finally, this is a very rapidly moving field. I think as any of us working in it are all familiar with, there are constantly being new publications of new genes that we ought to be including. So you have to have the flexibility of panel design to be able to say, right, I'm now ready for version 2. Can we now add on some new genes onto my existing panel? So on the next slide, I kind of tried to summarize the things that we need to think about. So which genes and mutations do I need to sequence the whole gene, or can I just focus on some hotspots? And what kind of mutations do I want to look at? Now, traditionally, we've used next-generation sequencing to look at SNVs, or single nucleotide variants, base changes, missense changes, nonsense changes, and some small insertions and deletions. But in fact, a lot of the mutations that we're asked to look at in these tumour samples are actually copy number changes and gene fusions or translocations, like the EML4 ALK gene fusion. So can we actually identify these kind of mutations at the same time as our single nucleotide variants? The choice of different library preparation methods is growing all of the time, and there are lots of different library methods which ought to be available to you, depending upon which particular company you work with. And some are better and some are worse for FFPE samples. You need to think about the coverage that you want to achieve. So this is both the lateral coverage in terms of the spread, the width of the genes that you want to analyse, but more importantly, the depth of coverage. So how many times do you want to sequence each of your amplicons? And this will in turn determine the sensitivity and the specificity of your assay. And in these kind of clinical situations, we really don't want to miss anything. We need to develop an assay which is going to work most of the time on most of our samples, even though a large number of our samples are really of very poor quality. And then going back to flexibility, are we going to have a panel which is going to be absolutely locked down, or should it be bespoke, where you have the ability to go back to your panel provider and say, I now want to add on an additional gene? So now on the next slide, I'm going to be telling you about some of the work that we've done on the Stratified Medicine Programme. So this is the CRUK StratMed programme using a Nextera Next Generation Sequencing lung panel. Now this panel consists of 28 genes, and included in those 28 genes are genes which we expect to see having altered copy number variants, and also gene fusions as well. And this is in addition to our regular SNVs. So what I'm going to talk you through is the way that we validated this panel. So we firstly designed the panel for the 28 genes, including the gene fusions we wanted to detect. We tried to ensure that we had good coverage of all the genes that we were interested in. Some genes we just covered the hotspots, and other genes we covered the entire coding sequence. So what we did first, and this is working very closely with Illumina, was that we actually worked on some cell line DNA samples to ensure that within cell lines we were able to detect mutations that we expected to. And the great thing about cell line DNA is that in fact you can create controls whereby you've only got quite a low mutation frequency. So we could really challenge the sensitivity of our gene panel. Now in this particular case, we were able to validate our translocations, so our gene fusions, but we didn't have sufficient clinical samples to really properly validate our copy number variants. We then transferred this technology into clinical laboratories. There were 3 laboratories involved, and we ran these Nextera panels again on cell lines, on previously analysed samples where we knew exactly what mutations we were expecting to find, and also on some real blinded samples. And then we looked at the concordance of results. So between our previous methods that we used, which might have been pyrosequencing, Sanger sequencing, or maybe even FISH analysis, and compared that concordance with the mutations that we were able to detect using next-generation sequencing. And as a guide, we gave ourselves a cutoff of 90% to say if we could get greater than 90% concordance, we'd be happy that the panel was validated and we could go ahead and use it clinically. And the interesting thing here, of course, is that, yes, there was the odd mutation that was missed by next-generation sequencing, although in every case we could come up with a good reason why, and that reason might have been poor quality DNA, or in fact the probe, in terms of for the library preparation, was sat in the wrong place and sat on top of a mutation. But there were also some mutations which weren't detected by our primary molecular analysis, so Sanger sequencing, but were detected using next-generation sequencing. And this really gave us a lot of confidence that we had good sensitivity using our next-generation sequencing. So if we move to the next slide, the wet lab work around next-generation sequencing, which we've talked a little bit about the design, is just half of the package. Another really big part of your next-generation sequencing pipeline is your bioinformatic analysis. And it's really important that you put a lot of effort into either using a commercial bioinformatic package or you're able to develop something locally. And the types of things that you need to be thinking about are, can we align these really short fragments that we're going to be working on from our FFPE samples? Do we need to run a comparison with our genomic sequence? So we found that when we were trying to detect copy number variants, we did need to have a genomic sequence control run from the same patient at the same time. Is the pipeline able to detect the variants that we want and also annotate them, so correctly name them? We wanted to be sure that our bioinformatic pipeline was able to recognize low-level artifacts, like the ones that I showed you earlier, and remove them, because we knew we didn't actually want to look at those. And also recognize recurrent variants and artifacts, which again we knew and we'd been able to validate as being technical artifacts. So we wanted the bioinformatics to ignore those. We didn't have to look at them every time. We wanted to be able to detect changes at around about the 5% level, or whatever, which is what we'd actually validated for numbers of panels that we use. We wanted to be able to use our pipeline to be able to detect duplicates. So this is where the same PCR fragment, shall we say, has been sequenced over and over and over again. We wanted to remove that because that was going to introduce bias to the sensitivity of the assay. So any fragments which had the same start and finish in terms of sequencing, we wanted to remove from our bioinformatic pipeline. Of course, we wanted to be able to detect copy number variants and translocations, and this was something that was really quite challenging because this was new to the kind of bioinformatic pipelines that we were using. And at the end of the day, we also wanted a pipeline that was relatively user-friendly. I only have one bioinformatician in my department. I couldn't call him in every time we wanted to analyze some next-generation sequencing data. I wanted all my scientists and technologists to be able to use this particular application. At the end of the day, we need to be confident that any variants that we're then able to detect through this bioinformatic pipeline were real. We're only looking at the significant ones. I'm not interested in looking at polymorphisms. We also weren't missing anything. We needed to have confidence that if a patient was wild type for a gene, it truly was wild type, and that any variants we detected had been correctly annotated, so they had the correct nomenclature. So on the next slide, I just have a breakdown of how our bioinformatic tool actually works. So it's really quite simple, and the first 3 steps are all now automated through our pipelines, where the raw data is outputted from the sequencer. That data is then analyzed in terms of alignment using a whole series of open source tools. We're then able to collate the variants which we believe to be real and significant for that patient. And then the final step, which is actually done manually, is the assessment of those variants to make a determination about whether we think that they're significant or not, and that would be done by a scientist. On the next slide, we have a schematic of the whole of the bioinformatic pipeline, and I don't expect to go through every step of this, except to say that it's very complicated, and it's really important that you have bioinformatic support in putting together these pipelines, so that the user input is only at the very late stages in the last few boxes at the bottom of that slide. So to show you a few examples of the kind of results that we're actually able to get out, particularly focusing on the copy number variants and translocations, on the next slide, we actually have the output for what we'd see when we're able to detect copy number variants. So SNVs, so the single nucleotide variants and the small insertions and deletions, hopefully many of you will be familiar with, and they're relatively easy to detect. To detect a copy number variant is far more difficult, and we've been able to achieve this by taking our solid tumour sample, so DNA extracted from FFPE, and sequencing both it and DNA extracted from the germline, and comparing the two and doing a subtraction from one with the other, which gives you the kind of imprint that we have, and then stacking up, as we have here, on a gene-by-gene basis. And what this is able to show us is that we have allelic imbalances, so losses of gains, in comparison with the germline sequence. And on this particular sample, you'll see that we've got an increase of MET up at about the, I can't remember, it's about the 4 or 5 times level, and a drop, in fact, complete deletion of the CDKN2A gene. And so this is the way that we're really quite easily, as users, able to interface with the bioinformatics to visually see whether we have copy number changes. Now, at the current time, for these copy number changes, we're then going on and validating them using another method. So we've chosen to use FISH, but equally you could use real-time PCR to give us more confidence around the sensitivity of the detection of copy number variants. And similarly, on the next slide, we have a different bit of software in our bioinformatic pipeline is able to detect gene fusions. It's only focused on particular gene fusions rather than any gene fusion, but our software is able to stack up the gene fusion so that it looks like this and says these 2 bits of the gene. So in this case, the KIS FFPE5B gene is brought together with the RET gene, and we're able to see that stacked up within our sequencing. So moving on slightly, let's talk a little bit about sensitivity now and the sensitivity of the detection of mutations. This is on the next slide. What I've tried to demonstrate here is the fact that your massive tumour cells is a complete mix of different cell types. So if we were to think about EGFR mutations, within our normal lung cancer cells, in our normal lung cells, none of the cells will actually have EGFR mutations if we're normal, if we're well. However, if we take a ball of tumour cells, what we actually have is a mixture of tumour cells, which I've shown in blue here, and normal cells, which are shown in white. Now, if we extract DNA from the whole mix, we're only going to have a proportion of that DNA is actually going to be mutant. So in this case, if we were looking for an EGFR mutation, we would only find those EGFR mutations in DNA which has come from the tumour cells. So the amount of mutant DNA that you expect to see is always going to be proportional to the mix of cells from which you've extracted your DNA. So how many normal cells do you have and how many tumour cells do you have? And we found that we need to factor this information in to our next generation sequencing pipeline. So on the next slide, we've got some rather pretty pictures of some trees. We also need to consider if the tumour contains subclones. So it's not good enough that that tumour is a mixture of both normal cells and tumour cells. We might find a subclonal population within the tumour cells as well, because a particular mutation has arisen later within that particular tumour. And this information was quite elegantly described in New England Journal of Medicine in 2014. But it's very important we can detect subclonal mutations as well as mutations which might be throughout all of the tumour cells. So on the next slide, we need to really think quite carefully about heterogeneity and how deep do we need to sequence in order to detect a mutation which may only be present in a proportion of our tumour cells, so it might only be present in a subclonal population. But we also need to be able to have confidence that if we sequence a sample and we do not detect any mutations, so that sample is truly normal. And we're able to do this by relating back our sequencing depth to our estimate of neoplastic cell content within the sample in the first place. So it's really important with each of our pathology samples we have a really accurate assessment of the percentage of tumour cells within the DNA sample which we've extracted and then we're submitting for next generation sequencing. So on the rather busy next slide, I've tried to run through this. And for those of you who aren't familiar with next generation sequencing, the number of times each gene or gene fragment is sequenced is known as the depth or coverage, and that tells us how many reads we have, or sequencing reads, of each particular gene sequence. And because we have variable numbers of gene read sequences, so that relates to our depth or coverage, that's what makes next generation sequencing a quantitative assay. So if we had a sample in which we had pure tumour cells, no contaminating normal cells, just pure tumour cells, so 100% tumour, and we knew that that had a KRAS mutation, which was just present at 25%, so it was a subclonal KRAS mutation. If we then sequence that sample and we're able to detect 100 reads, 75 of those reads would have the G base, which is the normal base, and 25 of the reads would have the A base, which is the mutant base. So it allows us, the technology really allows us to quantify these very low-level mutations and look for needles in haystacks. Now, however, we know that our next-generation sequencing is sensitive to around about 10%, and we know this because we submitted, as part of our validation, samples where we knew we had mutations at 10%, at 5%, 20%, 50%. We did the full works. So this would mean that if we had, again, a tumour sample that was absolutely pure, 100% tumour cells, and we had 100 reads across a particular gene, that we ought to be able to detect a variant if it's present at the level of 10%, so 10 of those reads out of 100. However, it's worth bearing in mind that we never have samples which are 100% tumour cells, and they're generally always less than 100%, sometimes considerably so. So as we decrease the amount of tumour cells that we put into the mix, we either have to increase the level of sequencing that we're performing to keep that sensitivity the same, or we need to, increase the read depth in order to do that. So on the next slide, I hopefully have demonstrated this to you, where we have a table of the number of, down the left-hand side, the neoplastic cell content of a particular sample, going from 5% all the way up to 100%. And then if a variant was present just at 10%, so it was a variant that we saw in a subclone, you have the frequency that you would expect to see that variant then, and therefore then the number of mutant reads you would expect to see, and therefore if we're to detect 10 mutant reads, what kind of read depth would we need to achieve? So if we take the third line down, where we have a neoplastic cell content or tumour cell content of 20%, if we had a mutation that was present only in 10% of those tumour cells, we would expect the total amount of mutant DNA to be down at 2%. So if we want to detect that mutation at 2%, that means 1 in 50 reads are going to be mutant. We know we've got to detect 10 reads to have the confidence that that mutation is actually there. So we actually need to have a read depth of 500. And a read depth of 500 is really quite high in terms of the sequencing capacity. But given that most of our tumours are around the 20%, 40, 50% neoplastic cell content level, that's the kind of read depth that we really need to achieve. So I've tried to demonstrate in the next slide that the way we use this is within the CRUK Stratified Medicine programme. So where we've determined that to actually detect mutations is probably the easiest thing, but determining that a sample is normal is far more difficult. So what I've shown here is the confidence that you have having, so if you know the neoplastic cell content of your tumour, you know how many reads you actually have, and how much confidence you can have if you haven't detected any mutations, that that sample is actually wild type over a particular gene area, a particular region of interest. So ROI stands for region of interest. So this would mean if you took a tumour which had a tumour cell content of 60%, so that's the middle line, and you had a read depth of 167, this would tell us that we actually had high confidence of detecting a wild-type mutation should it be present. So we're able to combine the number of reads that we see and the tumour percentage to be able to give us the confidence that the sequence has been completed satisfactorily, which kind of gives us a quality score. And I think this is really important, particularly when it comes to determining that a particular gene sequence is wild-type. So on the next slide, we have your second poll question.
Great, thank you very much, Rachel. And the second question for you is, can you estimate the percent savings that can be made by transitioning from single gene tests performed in series to sequencing panels? Answer A, 5%, B, 25%, or C, 40%. Again, can you estimate the percent savings that can be made by transitioning from single gene tests performed in series to sequencing panels? Answer A, B, or C now, please. Okay, great. So we have the highest number is 74.3% of people have answered overwhelmingly for 40%. And we have 25% of respondents have answered 25%. Rachel, what do you say to that?
Yeah, now I think this is really quite interesting. I'd have gone for option B at 25%, but of course there's an awful lot of factors here that you'd have to take into consideration. But it's great to think that people do feel that you make good savings by using this particular technology. It obviously does depend upon the number of genes that you're analysing and the size of the genes. It depends upon the cost of both technologies. So, you know, we're using a particularly expensive technology previously for your single gene tests. And then, so if you're using something very expensive like fish, and then you're then able to get away with a fish, then you're going to make even better savings. And I think the other thing that we always need to remember is that we tend to focus with next generation sequencing on the cost of the reagents, because they're the expensive bit. It's also worth remembering how much staff savings you're making. If you're not having to run a whole series of molecular tests in series, you're going to save a huge amount of staff time, particularly if you've managed to nail that bioinformatic pipeline and got that working really well. So I'm now just going to move on, just for the last few slides, to talk about some of the other applications of next-generation sequencing for solid tumours. So we've demonstrated the next-generation sequencing gene panel can be used for our paraffin-fixed samples, and hopefully we can use it really quite effectively. However, we also know that once we've actually delivered our molecular test on the patient's primary tumour, we've got our molecular result, and the patient is receiving their personalised or stratified treatment, that eventually, after a period of time, that patient will stop responding to that treatment which has been selected for them. And we know that patients stop responding because they actually start to develop resistance mutations. So this goes back to the subclonal nature of our tumour. So those small subclones start to grow out and causing the patient to be resistant to the treatment they're receiving. So as we think to the future and the way we're going to be delivering our clinical services, we need to start thinking about how we're actually going to detect some of these resistance mutations and what kind of technologies might we be using. The problem with some of these new mutations is that they're always going to be at a low level, so we do need to have really sensitive technologies. Now, one way that we can look for these resistance mutations is to use circulating tumour DNA. Now, the benefit of using circulating tumour DNA is that we don't have to go back and take a new biopsy from the patient, which in many cases the patient is not going to be happy about us doing. So if we can take blood samples and then extract circulating tumour DNA from the patients, that's able to give us a longitudinal sample, so we could take those samples every 3-4 months, depending on the particular tumour type, and analyse those circulating tumour DNA samples. We should hopefully be able to detect the molecular mechanisms of resistance, which should then enable us to determine what the next best treatment for that particular patient is. On the next slide, we've got an example of this for the Focus 4 clinical trial, which I showed you before. But in this particular trial, we're actually taking blood samples and extracting the circulating tumour DNA initially, so when the patient is first recruited to the trial. But then, as they then start receiving treatment through the whole process of the clinical trial, we'll take longitudinal samples so we can start to map when that patient might start developing resistance to treatment. Another application of using circulating tumour DNA is on the next slide, is if we go back to EGFR, which we're all familiar with, we know that a particular mutation, the T790M mutation, is a mutation which causes resistance to EGFR TKIs. And there are now beginning to be agents on the market, which hopefully will be licensed soon, which are going to be specifically for the use in those patients who have a resistant T790M mutation. We know about 50% of lung cancer patients will develop resistance due to the T790M mutation. And these patients will respond to these third-generation EGFR TKIs. So on the next slide, we have a cartoon of where circulating tumour DNA comes from. So essentially, it's DNA that has been shed from the tumour into the bloodstream. Now, the problem with circulating tumour DNA is it can often be there in very, very small quantities. So just as little as, well, less than 1% or 1 to 2% of the total circulating tumour DNA has actually come from the tumour, or the total circulating DNA, free DNA. The actual amount of circulating tumour DNA is often associated or correlated to the tumour burden, but it does seem to vary hugely between patients and also the treatment point or the treatment time of those particular patients. So what we need to focus on is what technologies can we use for the analysis of this circulating tumour DNA. So briefly on the next slide, we have some of the pros and cons of the analysis of paraffin-fixed tissue compared with circulating tumour DNA. And the real problems, which we've already talked about, of analysing paraffin-fixed tumour DNA are the quantity of DNA that we're actually able to extract and also the quality of DNA. The fact that the turnaround times can take such a long time because of getting the samples from pathologists, which again we've already talked about. And some patients actually have no tumour sample available, particularly when it comes to looking for resistance and the possibility of rebiopsies. And I think this is really where circulating tumour DNA can be beneficial. However, we do need to bear in mind that only small quantities of circulating tumour DNA are extracted, and we have to handle these circulating tumour DNA samples very carefully, because the circulating tumour DNA will start degrading very rapidly after that blood sample has been taken. However, having borne those things in mind, there is really quite a lot that we can do with circulating tumour DNA. So on the next slide, I've been able to demonstrate the way that we've used next-generation sequencing now to be able to detect mutations from circulating tumour DNA samples. So if we actually look at the axis of the graph that you can see on this slide, you can see that we've got considerably less target DNA than we have when we were using our FFPE or our paraffin-thick samples. So now we have to sequence at a much higher depth in order to have the sensitivity that we need from this particular technology, because we're looking to detect mutations at around about the 1%, 2% level. And you can see this particular patient who's had circulating tumour DNA samples taken at 4 different time points. We can see 3 different mutations which are increasing with this patient's treatment. So as the patient starts progressing on their treatment, the amount of circulating tumour DNA, as seen by these 3 molecular biomarkers effectively, is increasing. And this correlates with the treatment response of the patient that you can see in the scans down below. So in the first set of scans, you can see that the tumour is relatively small, and in the second set of scans, that lesion is now growing, which correlates with the increasing amount of circulating tumour DNA. So if we think about the future of molecular pathology services now, and we go back to our original, well, what are we trying to deliver in terms of patient stratification? What we want to be able to achieve is to deliver gene panel testing for patient stratification so that we can, at the same time, we can get patients directed to the right clinical services and clinical trials. We also know we need to have really great relationships with our pathologists to ensure that we maximise the patient samples that we receive and we're able to work on. And we need to be thinking about future strategies with our molecular methods for how we're going to predict the patient's relapse and resistance maybe to the treatment that they're receiving. So finally, and in summary, I hope I've demonstrated that we can use next-generation sequencing on paraffin-thick samples and also circulating tumour DNA. It is technically demanding, but it's generally limited by the sample quantity and quality. Those are the things which make it difficult. There are, however, an increasing number of commercial and bespoke products that we can use on the market for our next-generation sequencing, and you need to be quite careful about what you're going to select in terms of the right panel for you and for your application. And it is also really important that you validate the whole of your analysis pipeline, from the design of the gene panel, your wet lab work, and particularly your bioinformatics. So I hope that's given you a flavour of what we're doing in Wales with our next-generation sequencing, and I'm very happy to take some questions.
Thank you very much, Rachel, for this excellent presentation and for relaying your experiences to our audience. So yes, as Rachel has said, we are now ready to start the questions and answers session. We do have some that we have received, and like I said earlier, please feel free to submit questions as we go. We'll try and get through as many as we possibly can in the allotted time. But please rest assured that any questions that we don't have the time to answer live today will be answered via email. Okay, thank you very much. So let's start with our first question then. Rachel, we have a question here that's asking, what do you think of variants of unknown significance, and how do you think they should be dealt with when they arise?
The way that we handle them here is that we're actually quite careful to avoid any variants of unknown significance if we can in the first place. So if we've been asked to perform an analysis of 5 genes, but there are 20 genes on a gene panel, we'd only analyse those 5 genes. So the first thing is to try and limit the variants that you really don't want to be bothered with and that might cause you difficulties. The patient's only be consented for analysis of your 5 genes, so why would you look at anything else? So that's the first step. Then in terms of if you do detect a variant of unknown significance in the gene that you're that you have been asked to analyse, then it's a matter of having a really good conversation and communication with your referring oncologist and ensuring that they understand what you've found. We would go through the process of looking up that variant in the literature. Has it been seen before? What does in silico analysis about that particular variant tell you? Does it tell you whether it's an important change at the nucleotide or the amino acid level? Might the particular change predict a splicing change? And then we would share that information with the referring oncologist to give them as good an understanding as we have, and hopefully help them to make their treatment decisions. But at the end of the day, these variants are going to be very rare, and it's going to have to be down to the oncologist to make their decision about how they're going to treat the patient.
Great, thank you very much for that. And another question that's just come in. What's the minimum amount of DNA you need for NGS testing? The person that's asking the question says that he's been told that there are technologies that only need 10 ng or less and still provide good data. Have you tried this?
Yeah, he or she are quite right. It does depend upon the library preparation method. So different methodologies require different amounts of DNA, and that also relates to the size of the gene panel as well. So the larger gene panel, the more sequencing that you're trying to do, you will need to put more DNA in the first place. But it does depend upon the library preparation method, and he's quite right that there are methodologies, really very good methods out there, which will go down to as little as 10 nanograms, which is really very useful when it comes to the kinds of samples, some of the tiny biopsies that we're actually handling. Many of the other methodologies might use 30 or 40 nanograms, but I think the commercial companies are really doing a great job at the moment in recognising that this is difficult for us to actually get the amount of DNA which is required.
Great, thank you. And another question, the person is asking, how do you actually isolate circulating tumour DNA, and how do you actually distinguish between circulating DNA and circulating tumour DNA?
Right, that's a very good question. So there are commercial kits available for the extraction of circulating tumour DNA. So firstly, you only extract from the plasma. So the first thing you do is just spin down your blood sample to get rid of all of the lymphocyte DNA and the buffy coats. So you get rid of that. You're just left with the plasma, which includes the circulating tumour DNA, or the circulating free DNA as well. And there are various kits. I probably shouldn't tell you which providers there are, but there are various kits for actually extracting, very effective kits for extracting the circulating free DNA. The next question is very difficult. So how do you tell how much circulating free DNA do you have, which comes from your host through the normal apoptosis of cells, and how much circulating tumour DNA you have? Now, there are methods to look at this because circulating tumour DNA is said to be shorter than circulating free DNA, so the actual length of DNA is variable, but we haven't found that to be a particularly good methodology of discerning between the two. So we take the approach of actually looking at molecular biomarkers to tell us how much circulating tumour DNA there is, on the basis that if you can actually detect a mutation, I think as I showed in my slides, then that gives you a handle to actually start performing a quantitative analysis, which will tell you if that mutation is only present on the tumour DNA, that will tell you how much tumour DNA is present within the mix. But I do agree, it's quite a difficult area to get your head around to have an understanding of these differences.
Wonderful, thank you. And what do you consider the biggest benefit that NGS has actually brought to your laboratory?
I think it's really the fact that we can now analyse far greater numbers of genes simultaneously. So it's a resources issue. It's the fact that the analyses that we wouldn't have dreamed of before, we very much went one gene at a time, one exon at a time, and it was a very time-consuming manual process, particularly if you were screening for unknown mutations. So say you were screening something like the P10 gene or P53, that was a very slow and laborious process for us using the old technologies. But now the fact on next generation sequencing, it's far more rapid. We're not actually looking at sequence data anymore because that's all produced for us through our Bioinformatic Pipeline. So I see the major benefit is, it's a resources one. We can do far more with our money, with the funding that we have, which is great for patients.
Well, that's a fantastic one to end on, actually, Rachel. We've actually run out of time. Thank you very much once again. Thank you to Rachel. Thank you, everyone, for attending. Look forward to seeing you all at future webinars. Thank you so much, and goodbye.